All right. Hi, everybody. Welcome to week 3 of our webinar series, Microbiomes for All. I'm Rachel Horak, ASM staff. And today we're going to hear more about data analysis, looking at heat maps. And so all of your resources that you're going to need today have been posted to the LMS. If you didn't see them, go ahead and refresh your browser because it should be there now. And as we go, I'll put links in the chat. Okay, so, and one announcement, make sure that as we go after every webinar, you do a very quick 4-question survey. It's called a post-webinar survey. And that feedback is really valuable because we do plan on offering it one more year. And we always like to get the feedback to improve, make sure that we're hitting the mark. Okay, so with that, I'll turn it over to our speakers and I'll see you at the end of the hour.
All right, welcome back, everyone. Hopefully you guys had a good intervening 2 weeks. Let me see if I can share my screen here for a sec. Okay. Can people see my screen?
Yep, you got it.
Okay. So I want to just, oops, if I can find what I want to see. So I wanted to just address one of the issues that came up at the end of the second session. So I'm right now looking at, I hope what's the Google Drive folder that everyone has access to, and the one that has the materials in it. And if we go to the Nepheli outputs, and if we go to the outputs for data 2, for the data 2 pathway, which is this folder, and we had been looking at some of these HTML files. Let me see if I can. Okay, so this is just an example of one, and some people were not having the same luck opening it quite like that, and we took a little time to try to figure out what was going on. But if you are one of the people that had issues opening those HTML files, maybe the message got out to you, but if it hasn't already gotten out to you, it should work. We tested this. If you download the entire graphs folder, it looks like there are some sub files within the graphs folder that you need to be able to open the HTML. And if you just download the HTML, people were not getting anything. But if you download the graphs folder, then you should be able to open up the HTML files within that. So hopefully that might address that issue that people were having. If not, we can continue to try to troubleshoot it. Okay, so so as we're moving into session 3, we're going to continue with the data analysis. And again, on this kind of big picture view, we're over here kind of looking at the various output files and how you can work with these and generate your own with your students. And I'm going to talk a little bit more about what came in the analyze pathway from QIIME 2. And then we'll move into the QIIME 2 Explore. And if everything works live fine, then I shouldn't need to use all the slides, but you'll see in the slide deck there are static images of most of the things that I'm going to be going over. And so if you remember from the Nepheli kind of workspace, we have the three major pathways, which are the pre-processing, the analyzing, and the explore, and loading your files and metadata is pretty much the same for all of these different steps. And we talked last time about the analyze pathways and the data2 pathway and the QIIME2 pathway. And if you run those, you will again get the automated emails. And then when the pipeline is finished running, you'll get the notification, and then you can look for the output folders. And so again, we've done this, and most of these output folders are here in the Google Drive that you have access to. And so I was just showing you again the data2 output, but if we look at where is it? The outputs number 9, the QIIME2 analyze demo. There are again a bunch of output folders, and who can remind me the first folder that they want to check if they have any issues? It's a question from last time.
You've got this right before.
Log file.
Log file, right, there it is. Congrats, log file. And again, for I think I might have showed you for the explore, to go on to the explore step, you need the .biome file, which is down here at the bottom. And for some other things, you might need the tree file, which is up here, but I can revisit that. But one of the other things that I wanted to show is that there are some files that you can use right in this output folder from the analyze step. And one is the taxonomy.tsv. And here let's see if I can. Okay, so these are the taxonomic assignments from your run. Okay, and again, you can, like I just opened it right there on Google Sheets, but I normally download these and then I open them in Excel. I'm more comfortable working in Excel than I am in Google Sheets. And another very useful one is, where did it go? Is okay. It's not here as I don't see it here as a TSV, but there is the repsec.qzv, and I'll show you that in just a minute. I thought maybe it was as a TSV, but I don't see it here. Okay. So these files that say QZV, I think we talked about this a little bit, and that these are viewable through the QIIME2 view. And so if you go to QIIME2 view, which I have open here, you can click, you can go to the relevant folder, and you can see that all those that are QZV are open. And I think last time one of the last things we talked about was looking at the bar plot. I think I showed some of that. The taxonomy QZV I just opened in Google Sheets, and the other one that I wanted to look at was the repsec.qzv. Okay, and so here are again the sequences that are used to represent each of the taxa that were identified in your sample. So this is the representative sequence that QIIME is using to represent the given taxa. Now, unfortunately, you don't have the information linked. You have the taxon information and the taxa file, and you have the sequences in the repsec file. So what I often do, and I'm going to switch now to Excel for a minute, is, oops, no, that's not what I wanted. Okay, can people see my Excel file?
Yep.
Okay, so here's the representative sequences file that I was showing you, and this is what you would get if you download it as a TSV and open it up in Excel. And here is the one with the taxonomy information that you can also download as a TSV and open up. And what you can see is that they have, each one of the entries has this ID number. Okay, the feature ID. And what I do is I sort both of those files based on the feature ID, and then I copy and paste the sequence information into the taxa sheet so that I now have this merged, and I can see here, if I look at the first one, it's some archaea, and then here is the sequence that was used to make that identification. And I'll come back to this in a moment, because a lot of times working with your students, you may, if blasting is one of the things that you like to have them do, or generating trees, this is a nice merged file to have, and it's really relatively easy to make. And then again, you can share it with your students for them to kind of do other analyses. And I put in here, I think that this is available in the folder, and I put a little note here that kind of describes the sorting that you need to do to, just so that the feature IDs line up in both the other sheets so you can combine them. And I'll just pause for a minute. Are there any questions that anyone has about this? Anything in the chat? Okay. All right. So that is, again, let's see here. Okay. So that's pretty much all the things that I wanted to talk about in the analyze output folder. And what I want to switch to now is to talk more about some of the outputs from the QIIME to explore pathway. And so that would be, again, moving from the analyze step to the explore step to do this downstream analysis using the QIIME2, where we uploaded the biome files and so on, and your metadata files and so forth. And oops, the output for those is in the outputs 11 explore folder. Okay. And so if I click there, I can see that, again, there's a lot of folders. Again, there's our favorite log file if there's any issues. And you'll see, again, a bunch of QZV files, which we'll look at in a minute with the QIIME2 viewer. And also in some of these folders, say if you, there are outputs for alpha diversity, like Faith's phylogenetic distance, and I think this is Pilu's evenness. And if you open these, again, there is the TSV file that you can download and open in Excel. And so I think last week I showed some graphs where I might have done like a t-test on some of the phylogenetic diversity data. and that's coming directly from this QIIME2 output, and I'm downloading, I can, oh, yeah, so you can see right here, here are my samples. Oops, something else opened here. Here are my samples, and then here is the Faith's PD score. So you can get these data directly and work with them in a spreadsheet to do whatever else you might be interested in if you want the students to have direct access to those numbers. So what I wanted to take a minute now to look at is we talked about, again, the bar chart from last week, and that is also in this folder as well. So there's my bar plot. It's at the domain level now. I can move to the phylum level. I think I might have rushed a little bit through this at the end of the second session. I don't know if there are questions on either getting this to show up or the things that you can do from here. I'm happy to kind of go over it again or address if there are questions or things.
Elaine was asking where did the data come from, and I just told her it's from the Explorer pipeline in China.
Yeah, so right, so this is in the Nepheli outputs folder. This is the Explorer outputs. So if you run the Nepheli Explorer pipeline here, you're going to generate this outputs folder. I've just named it Explorer Demo, and all of these files are going to be there. And what I just opened up is this one down here at the bottom, barplot.qzv, and I opened that through the QIIME2 view. Did that answer that question?
Got it.
Okay. And again, you can sort samples by treatment group, or again, this is all coming straight from your metadata file. And you can go, this is the phylum level. You can go down to genus if you want, but it gets kind of crazy there. The other thing that you can do is you can download these data as a CSV file, and then you can recreate the bar chart in your own Excel if you'd like. I think maybe you're seeing this now. I didn't point out that if you hover over each of the little elements in these 100% bar charts, it tells you what it is that you're looking at and what the relative abundance of that particular taxon is. Okay. And again, I think this is like really exciting, and it's one of the first data that you can really get to, because you don't actually have to run the Explorer pipeline. This bar chart is also coming directly out of the Analyze pipeline, which I can.
Theo, just a question from
Yeah.
her. How do we know which level means phylum, family, genus, etc.?
Okay, just give me one sec. So just to again, here's the Analyze, and here's that same bar plot folder. So level 1 is domain, and then level 2 is phylum, class, order, family, genus, species. I don't know why they do it with levels 1 through 7 instead of just putting domain, phylum, class, but that's the way it is. So.
And we have a second question from Laura. Can you filter out a genus?
So if you go to the level of genus, you can highlight. Okay, I think I can do it here. Yeah, you can highlight one, but you can't. In this view, you can't filter it out. That's again one of the upsides of using the CSV file, is that if you download the CSV file, then you could create a bar chart that is specifically, let's say you only wanted to look at the proteobacteria. You know, here I have my proteobacteria in green, and I can highlight it, but I can't drill down and look at, say, how are my proteobacteria broken up really easily. But if you do download the data as a CSV file, then you can do that. The only thing that I'll point out is that when you download it, it's best to download it at the level of genus, because the information at genus will keep the identities further up the taxonomic ranks. Whereas if I download it at phylum level, things are going to be clustered by phylum, and I'm not going to have those other finer breakdowns in the taxa. So I usually download at the level of genus. Does that answer the question? And after this, I'm going to talk some about Excel and show you a few things in Excel. And I can maybe, if there are questions about the bar plot there, I can go into that. Okay, so again, this is one of the first things you can get very, helpful to look at with your students. And again, you can see that here are samples that are divided by depth. So here are my shallow samples collected from 5 centimeters, and then here are a set of samples collected from 80 centimeters. And already, although I can see that they're quite similar and the same taxa are represented, I can start to ask, well, you know, it looks like proteobacteria are more abundant in the shallow samples, and the acidobacteria are more abundant in my deeper samples. And then you're off to the races with developing hypotheses or other things to kind of test those types of observations. Okay, so the other some of the other files that I wanted to show you, if I go into, again, now I'm switching back to the outputs from Explore, and I'm going to look at one of the Unifrac outputs. Okay, and so now I'm looking at a PCOA plot, and you can, it's always a lot of fun to rotate it. You can rotate this in 3 dimensions, and you can, let's see, you can start to color code these and look at questions about how are these different samples separating out. Okay, so again, I have my samples here in blue that were taken at 5 centimeters depth, and then my red samples here, my orange samples, which are more spread out, were taken at 80 centimeters depth. Now, if there are statisticians here, you probably fully understand, how these plots are generated and how best to interpret them. I'm not an expert statistician. I can't, recreate the generation of the eigenvalues and eigenvectors used to generate this information. But again, for our work with our students, we use this as a tool to show how different samples, if their microbiomes, are distinctive. And if you see clustering, that suggests that there are components within that microbiome that set that set of samples apart from, say, another set. So in this case, it's depth. We can look at, what do I have as sample type? So again, we have I have leaf samples here in red that are way over here on the right. The soil samples are kind of spread all over, and then my sediment samples are falling within the soil, which isn't too surprising, but they're kind of close together. So you can, guess that they might be pretty closely related. And again, depending on, you might want to look at different angles because, depending on how you look at it, it can be a little bit different. The other thing to point out is that the axes are representing kind of like the variation in these samples, and these three axes totaling together are not representing a whole lot of the variation in this set of samples. And so again, it's nice for, I would say demonstration purposes and illustrating the potential power of these types of tools. In this particular data set, it's not necessarily capturing as much of the variation as would be ideal. So again, it's kind of a good place to start in terms of generating questions and hypotheses, but it's not necessarily saying anything particularly significant about how these samples differ. Does that make sense to people? Yeah, question?
A couple of questions. Is this equivalent to PCA is the first? And then do you mind just reminding folks what the axes, each of the axes represents, if you can?
Okay, so it's not the same as PCA. I have to go back and check the definitions, but this is principal coordinate analysis as opposed to, I think, principal composition analysis. And if you look at, remember I was showing last time the StatsQuest YouTube tutorials, that's a good place to look for the distinctions on those. And I have to go back and check myself. In terms, oh yeah.
Yeah, the axes is the second one.
Yeah. So you do have a plot here that's kind of showing how much of this variation is represented on the axes. But this isn't the type of analysis that, at least from what we have available here, that really explains what is it that's represented on axis 1, 2, or 3. Okay. So for that type of answering that type of question, and I don't have it ready for today, I could do it next week, is using like the linear discriminant analysis function that isn't a part of the Nepheli pathway. It is in the microbiome DB. But if you do something like the LEFC or the linear discriminant analysis, that you can compare samples, and it will highlight the actual, if you're measuring taxa like we are here, it'll show you which taxa are enriched in one sample versus another. That's not too different from the bar plot that we looked at. That information is in the bar plot, but in the LEFC analysis, it's actually a little bit easier. The way it's organized is kind of easier to see that, but that you can't easily see in the PCOA plots. So if we can ask at the end of the session if people would like to see that. I doubt I'll have time to do it today, but we'll see. Other questions?
I think we got most of them. Jorge is just asking again if there's any, can we do statistics on this, or is it just for visualization? And it's pretty much just for visualization.
If you're a statistician, you can do statistics.
Yeah, we're just using for visuals.
We don't do that in the class, and in the research that we do in my lab, don't know if I've ever published with a PCOA plot and done any formal statistics on it. But I think, again, what you'll see if you look in publications is, again, and I don't know if it's, I don't know the statistical underpinnings of it, but what people will do is draw circles around samples, okay? And if you can draw a circle or, an ellipse around a set of samples and exclude this, include all the samples in one category and exclude the samples in another category, then that's oftentimes presented as evidence that they are distinct. But like I said, the statistics that would justify that is, I'd have to go and read about it because it's not kind of in my normal field of expertise. For students, it's nice to just visualize. You'll notice, so this is unifrac unweighted. So unifrac, remember, well, I don't know if we've talked about it before, is a method that basically uses branch length in identifying the diversity within a sample. And I was looking at unweighted, right?
Yep.
There is also weighted, and again, doesn't look, well, it didn't really color it, but when you look at, oops, this, again, the general take home is the same. The difference between the weighted and the unweighted is that in this case, weighted means that the abundance of each of the taxa, so not just the branch length that was used to kind of determine the diversity within a sample, but also the abundance of elements with that sample are factored in. Whereas the unweighted is kind of a binary yes/no, like was taxon 1 and taxon 2 present, or were those taxa absent? And so the unweighted doesn't take into account the relative abundance. So just in case you were curious about that. Okay. So I will now move on. And oops, what I want to do. The last thing that I'll talk about in terms of Nepheli, and this is the pie crust tool. And I don't actually, let me just check. I don't think I ran this, so I don't think that there's an outputs folder for this. Let me just double check. Oh, no, I guess I did. All right. So for the pie crust, what this does is essentially allows you to get an idea of what the metabolic potential is within a sample. And so what it does, kind of in a nutshell, is to take all of the taxa IDs that you get and matches them to a closely related genome that's available in the database. And then it says, so if you have A and A prime, the genome for A prime is in the database, then we're going to guesstimate that your genome is going to be very similar to A prime, and we can use that genome to represent the genes that are in your sample. And so it's oftentimes described as the poor person's metagenomic shotgun sequencing, because you can get at all of the metagenomic, or all the genes that might be present in the metagenome using this process. Like the other pathways, it's the same process. I won't run through it because I've done it already a couple times, but you're going to look for your biome file and then your metadata file and then a tree file, and then you upload it and then the outputs come here, and there's again lots of data within these, but the quickest one to look at, and again, if you're having trouble with the heat maps, I don't know if that'll happen for you. Try downloading the whole folder. And so here's the pie crust output, and again, my samples are here across the top, and this it's not really the first row, but this row here, the different metadata categorizations are up in these rows here. So I have my treatment group, my depth, all that is up here. And then here you can see all of these different metabolic pathways that are represented within the sample. And I think there's a way, I have to double check, that you can look at either the Kyoto or where was that function? All right, I'll have to double check. But this can be, again, if you're going into more of looking to identify, if you have a sample that was collected under anaerobic conditions, or if you have samples that were collected at, say, a heat-stressed environment, here's where you can go and dig and say, okay, we collected these samples from a heat-stressed environment. We would expect that the microorganisms that are abundant here are going to have enriched genes for protection against heat stress. So they might have more heat shock proteins. And so then you can go through the data to look for that. Or if they're anaerobic, or you can look for genes that are involved with metabolism in anaerobic environments. To be honest, we don't have time to get to this in the micro class, so I haven't generated any really useful tools for it, but I want to make sure that you know that it's available, because if you're doing anything with biochemistry, or if you want to talk more about metabolic pathways, that data is coming out in your analyses. Yeah, question?
A good one from Zamina. Is there a key to understand the color somewhere?
Yeah, so I was looking, and I feel like, I don't know if I can't see it there, because on my screen I've got a bunch of windows open. I felt like there was a key that kind of, I mean, when you highlight over these, it gives you the number.
Yeah.
And then you can go in and kind of look to see how these scores are calculated. But I thought that there was kind of like a little.
I thought there was too, and I don't know where it is.
I can't see it now. But yes, the basic answer is yes. And you can go and use these filtering. So I'm looking at everything now, but you could filter by like looking only at the soils from 5 centimeters or 80 centimeters and so forth. If this turns out, we can ask at the end that the people would like more with this, we can build it into the last session. But I just wanted to make sure that you knew that it was there. All right. Okay, so what I wanted to talk a little bit now is some of the tools that you can use in Excel. And again, another way to generate heat maps. So this is a Morpheus heat map that's created for you automatically, but I'll show you how you can use other data. And so just to kind of place us again, the data that I'm about to show you in the spreadsheet is coming from the QIIME2 outputs.
So just so he's on know, he's working in his drive. You just can't see it right now.
Oh, sorry.
All right,
I
no.
apologize because it shows up within the highlighted area, and I'm sorry, but I'm in the outputs 11 Explorer folder. This is the output from the QIIME2 Explorer pathway, and I'm opening that bar plot again. And those the data that I'm about to show you in the Excel spreadsheet are coming when I download this CSV file at level 2. Okay, so that's where it comes from. And I don't know if you saw that little pop-up because I guess you're not seeing any of the pop-ups, but that just went straight into my downloads folder. Okay, so let me switch here. Okay, can everyone see my spreadsheet?
Got it.
Okay, so here are what those data look like that get downloaded from the QIIME2 Explorer pathway, and we'd like to get to heat maps that look like this. And so I'll try to walk you through this quickly, and if people have questions, you can jump in. I put like a little bit of an explanation here, and I think we have a video of this on the website. But I'll give it a quick run here. Hopefully you'll be able to see what I'm doing, and there won't be too many pop-up windows. But I'm going to select everything here. So again, just to orient you, here are my sample IDs. Here are the different phyla up here in the first row. And then over here is my metadata. And I've just selected everything. And oops. Okay, selected everything. Actually, I'm going to create a new sheet first, and I'm going to call it demo here. I'm going to copy. I'm going to go to my demo sheet, and then I'm going to choose paste special. And again, I guess you probably can't see my pop-up window, but what?
You can see it this time.
Oh yeah, so I don't know why. Okay, so if you can see it, there's an option to check transpose, and I'm going to choose the transpose and paste. And so this is just flipping. So now the rows become columns and columns become rows. And so here are my taxa, and here's my metadata. And because those original file names aren't particularly helpful, I'm going to just copy over what they actually, the samples that they represent. Okay, so again, just so everyone's oriented, here in the first column are the taxa that are represented. Here across the top in the first row are my samples that we submitted. And then these numbers represent how many times was a sequence, in this case for Bacteria counted in the Prospect Park Lake. Oops, sorry. In the Prospect Park Lake #2 sample. So one of the first things, this is not necessarily the most helpful to have all the information, because like I said, when you download from QIIME2, it's going to bring along, in this case, the domain, and I really am only interested in the phyla. So I'm going to take my phyla, copy, paste it over here, and then I'm going to use a function from my data menu that says text to columns. And so if you're familiar with text to columns, great. If not, it's a really useful tool. So when I say text to columns, can you see the pop-up window here? Okay. It's going to allow me to break this data up into segments. Okay, so you want this delimited to be checked. And I just did this before, so it's defaulting to other with my underscore. And so what it's going to do is break that everything into a different column. Okay, and if it was like tab delimited or semicolon delimited, you could choose that as well. But here I want to break it down by those underscores. This is just asking me where do I want it to put it, and I'll have it just put it right where I pasted it. And so when I say OK or finish, here are now all of my phyla. You'll notice that one it was only able to identify to domain. So I'm going to just manually say that that's unknown. And so now I have all my phyla, which I can copy and put back here. So it's a little more friendly and I don't have all this other information. And I can go here and I can get rid of that. Oops. Okay. So if I want to start to make this look like a heat map, what I want to do is use conditional formatting. And so I'm going to select everything here. And again, I'm in a Mac Excel, so it may be different for you. But if I go to conditional formatting, I'm going to choose color scales. And there's a number of ways to get to the same place. I'm going to choose more rules here. And I'm going to choose a 3-color scale. Hopefully you're still seeing the pop-up here. And I have an option for a lowest value, and I'll make my lowest value blue. I have an option for my highest values, and I'll make my highest value red, and then I'll make the midpoint white. Okay. So if I do that, now it's starting to look a little bit more like a heat map, but I can still make some improvements here. So what I will do here is across the top, I'm just going to change this a little bit to make those look easier. I'm going to select again the area of my heat map, and I'm going to I think it looks a little bit better if everything has a border. And then I've got colors, which is going towards the heat map, but I've also got the numbers, and I'd like not to see the numbers. And so if I go into my formatting menu here for the cells, I have an option for more number formats. And if I go down here to custom, I can get rid of this general. And if I use this 2 semicolon selection, that tells it to get rid of the numbers. Okay. And so those, you can see up here in the formula bar that the numbers are, it's not like it's erased anything. It's just that you can't see them. Okay. So again, moving progressively closer to, oops, I don't want to do that, to what we would expect for a heat map. I'm going to select all my columns. I'm going to go to my format columns width. And I think 3 width of 3 kind of makes it look reasonable. Okay. So here, not bad. It's kind of looking like a heat map, right? And I'll show you a couple of tricks that I think can make this improve a little bit more. So I'm going to just copy the heat map area. Okay. And I got to resize my columns. Okay, so this is now more or less identical to the first, oops, not there, to the first one we had. But what I'm going to do is in the cell, okay, the value originally was 12, but I'm going to log transform the data. So I'm going to say equals, oops, log, and it's prompting me what number do I want to take the log of? Well, I want to take the number of the corresponding cell, which is here B2, and then the base. So we're not going to do log base 10, we're going to do log base 2, and enter. Okay, so now the value here is the log base 2 transformed value of the original one in the cell, and I can copy this and paste everywhere. And okay, we got some goodness and we got some badness here. It's got more of a range of colors, which is great, but I've got all these hashes which are coming up because I can't take the log of a 0 value. So all my zeros are coming up looking with those hashes. So the easiest thing to do, and I believe it's an accepted method, is I can just add 1 to each of my values. So every single value is going to have 1 additional count added to it. So that means my lowest value is going to be 1 instead of 0. So I'll do +1 and copy that. And there we go. And so now that looks like, I know Davida loves this, looks like a nice heat map. So a couple things to point out is that these were already sorted, I think, by the abundance of whatever the taxa were, okay? But, and so that's why all the red is for the most part at the top. Now, if I select everything, now, it's important that I have to go back to select from the original table as well, because these numbers in the second table are based on the ones in the first table. But if I select everything, oops, which I think, no, I guess I didn't. Okay, I'm going to try to select everything again. Why is it not? Okay. And I'm going to go up to data and to sort. And again, this is another nice function for the students to learn. It's really helpful. I'm going to say that my column has headers. I want to sort by, oops. No, I don't want to sort by that. All right, my thing is freezing up a little bit here. I want to sort by oh, there it is. Okay, index. And I want to sort from A to Z and hit OK. So now it's sorted alphabetically by phyla, and you can see that you know, it still looks like a heat map, but you don't have the kind of everything that was abundant clustering at the top. It's important, I think, and maybe you don't want to do it on such a massive heat map when you're trying to explain this, but in order to see patterns in data, it's very important how your axes are organized. And so you'll see in a lot of publications where the axes are ordered as a phylogenetic tree, or like we had the data on sample type. So if you're looking for trends, you don't want your data sorted alphabetically or anything like that. You want it to, you know, you want to manage those axes in a way that makes sense. All right. Any questions out there on the heat map? I think you're muted.
We just had one on adding the +1 for all, but I answered that one, so we're good.
Okay. Yeah. The one other thing that I'll point out is that when I okay, I'm going to select this again, and if I go back to my conditional formatting, and again, I'm doing this through the color scales, more rules, 3 colors, lowest value, blue, highest value, red. middle value white, the percentile, I'm not sure I fully understand this, but oftentimes it's better if you actually put in a number. If you look at your data and you see what is actually the midpoint, and so I did this before, so the midpoint for my log transformed values, I think, is 7.35, I think. And you'll notice it's not dramatic, but it does make it look even a little bit nicer than it was before. And so sometimes some little additions like that can be helpful. Okay, so a couple other things that I'll show, and the next thing that I like to do with the students, if I can move on if there aren't any questions, is a Venn diagram. And if this looks familiar, it should. This is exactly the same transposed data that I started out with for my heat map. So no difference, same data. Here are my phyla in the column A. Row 1 has all my samples, and the abundance of each one is shown in the cell, okay? I've simply copied over kind of the first column and the first row of that table. And what I've done is put in a formula. So if you can see, so here in the original data, the value there for leaf litter and cyanobacteria is 12. And the formula I have in my second table is, what I'm telling it is that if the value of B2 does not equal 0, then spit out A2. And A2 is cyanobacteria, okay? And if it is equal to 0, then just give me a 0. Okay, so that means that any non-zero value or any value above 0, what you're going to get is the name of that phyla back, and any place where that phyla was not present in your sample, you get a 0. So you get a table that looks like this, which isn't particularly useful, but if you go to one of the Venn diagram sites, and let me switch to that. Can people see my screen again and the Venn diagram?
I see the Venn diagram, yep.
Okay, so you'll see that it has lists, okay, and I can put, oops. And it's simply going to make a Venn diagram based on whatever information that I put in there, okay? And so I know you can't see my Excel file, but I'm just going back to the Excel file before, and I'm selecting that first column, say, for leaf litter, copying that column, and pasting it in, and I'm going to call this leaf litter. I'm going to go back and grab one for soil. Paste in one of the soil ones. And I'll grab one for sediment. And all I'm doing is grabbing those names from the columns I just generated. And I get a very nice Venn diagram. And what's particularly nice about this is, if you're interested in, say, well, is there a shared group of bacteria between these samples? Or if you're thinking that there might be like a core microbiome in a set of samples you have, if I click on the numbers here, down here, it tells me these are the common elements of these 3 samples that I entered in here, okay? And so I have a list of them, and I say, okay, well, here are my phyla that I think are shared among all of my samples. I could click here and see here are the ones that are unique to soil. Here are the ones that are unique to sediment. So not only is it kind of a nice diagram, but you can also get some very useful information because it's actually really cool to be able to pick out where those overlaps are. And again, I can add a fourth group if I want, and I'm taking that all from that table that I made in Excel. All right, so I think I'm running a little bit short on time. I wanted to see if there were any questions on what I showed for the heat map or the Venn diagram. The one other thing that I'll tell you is that same that same file that that same sheet that I created for the Venn diagram, you can go online and go to WordArt or any of the word cloud things. And obviously, like a word cloud is not going to be a really, something that you're going to put in a peer-reviewed publication. But for student posters or for students, wanting to visualize their data, word clouds are great. And you can, paste those things right into a word cloud generator, and this time include the values and where I was kind of omitting them. And you can generate, nice word clouds that show the relative abundance of the different taxa within your sample. So another really easy and fun visualization for students to do. So were there questions?
I don't see any in the chat that haven't been addressed, but I'll put the invitation out there for people to either write that question or come off mute.
And one other thing, before I forget, so hopefully everyone can, this is going to be in the slides. Next week, we're going to focus on nanopore data. And the nanopore data, you can run it through Nepheli, but it's really not, Nepheli is not designed to work with the nanopore data. And because of the low Fred scores, much of it gets filtered out. And so it's not a good match. But for any of you who have already worked with nanopore data, you'll know that Oxford Nanopore has its own analysis platform called Epitome. And next week, we'll talk about using Epitome Labs. And it's a really great tool for analyzing 16S microbiome data. And the reason I'm showing this is that if you want to play around with this, or if you want to follow along in our next session, you can go to the Nanopore site and go to this link and download Epitome Lab. And if you here's their website, you can if you go to the download, it's going to ask you to download. I'm on a Mac with a Apple chip, so I'm picking that appropriate download. You can pick the one that works best for you. Just a couple notes on that. If you're in the Mac environment and you don't already have Java, you'll need to do that, but it walks you through that. And you need another program called Docker. But both of those, it tells you that you need them, and it gives you you just click and it downloads automatically. So I've done it both on my desktop and my laptop. It's very straightforward. If you're doing it in Windows, you need to install a Linux environment, and my lab has done that, and also it walks you through all of that. So you don't need to do this, but if you want to kind of have it and play around, if you do this before we meet in 2 weeks, then you might be a little bit ahead of the game. And if you email us or Rachel, I know that I didn't show you a lot about pie crust. We could build some of that in if people are interested. If there are other things I didn't show you, I didn't show you the scatter plot in Excel or the rank abundance curves, I can show those. Those are in the spreadsheet that I put in the documentation, so you can look at it. But if you want me to run through it, I can also try to squeeze that into the last session. But just let us know so that we're kind of providing what people are interested in. Any other questions out there? I think you're muted.
Feel free to raise your hand and unmute yourself. I don't see any questions in the chat.
Okay. And like, you know, if questions come up later, don't hesitate to email myself, or if you email Davida or Rachel or Gina, they'll get in touch with me, or they'll answer the question themselves. Yeah, but I feel like I just ran a marathon. It's kind of a lot to get through. And again, as I've said before, the best thing to do is to kind of play around. And I know it's frustrating when you get stuck, but we're here. I'm on email almost all the time. And so I'll try to respond as quickly as I can if you find yourself getting stuck, and we can hopefully get you over those hurdles. But there's no substitute to just, you know, working with the files, working with our files, working with your files, and playing around with it.
Excellent. All right.
All right.
Yeah, no more questions. I'm going to stop the recording.